Welcome, everybody. Thank you for joining us for another presentation from the San Diego Rowing Club. On this, we're going to update you on COVID-19 with SDRC member Andy Geall. I'm Dan O'Neill, president of SDRC, and I'll be your moderator for tonight's webinar. Andy is a 20-year veteran of biotech and is currently the chief science officer of Precision NanoSystems. He previously worked for Novartis Vaccines, where he led and pioneered the exploration of mRNA vaccines. He holds 20 patents and has more than 20 publications under his name. He is an expert in his field, and tonight he has graciously volunteered his time to provide us with a scientific update on the state of COVID-19 vaccinations. So, a few things about our webinar. Mr. Geall will present for about 30 minutes, followed by Q&A from the audience. Within the Zoom interface should be a Q&A area where you can enter your questions, or you can press raise hand icon within Zoom, or put your questions in the chat. Andy has offered to answer as many questions as possible, so there's no hard and fast end time for tonight's meeting. Does anyone have any questions? Put your questions in chat or raise your hand. If you have, I don't see any questions. So let's begin. Andy, welcome and thank you for presenting this evening. I'll turn this over to you.
You're welcome. Thanks, Dan. So, everything just changed. So, here we go. So can you see that now?
Yes.
Perfect. Okay. So the first thing I'll say is by training, I'm a clinical pharmacist, but once I became a clinical pharmacist, I then went on to do my PhD, graduated from England in 1999. And my PhD was on making synthetic lipid particles to deliver DNA and RNA, and I spent my entire career pursuing that field, and I've done a lot of work in the RNA vaccine field. I'm going to present you some data from BioNTech, their phase 1, 2, and 3 studies, just because it's most likely that this is the vaccine you're going to get, or the Moderna vaccine if you get vaccinated. They're very similar, and I'll walk you through the different types of mRNA vaccine, because there's actually more than one.
Okay, Andy, yes. I think you're going to have to put that in presentation mode.
Here we go. How's that?
No, you need to put these. Yeah, there we. Let's. That's better.
How's that?
That's great. Thank you.
Okay, perfect. So I'm not an employee of BioNTech, but I've cited the references where I've pulled the data from. I'm certainly not a physician. I'm not going to be telling you to go get vaccinated. I'm here to supply you with the scientific information, allow you to make informed decisions yourself, or talk to your physician about whether to get vaccinated or not. But happy to, answer any of your questions and give you the relevant science that supports my answers. So currently I actually have 3 jobs. As Dan said, I'm Chief Scientific Officer at Precision NanoSystems. It's a Vancouver-based company, and we are working with the Canadian government. They funded us to take a RNA COVID vaccine into clinical trials, and that'll go into clinic towards the middle of this year. And with them, we're building a biomanufacturing facility that will be capable of responding to the next pandemic for the Canadian people. I also have my own RNA consultancy company. It's just me, and I've been consulting with the RNA vaccine field for the last 5 years. I've worked with the Gates Foundation. I sit on several scientific advisory boards. And I've also just recently co-founded a company in San Diego, Replicate Biosciences. It's a spin out from some technology at Duke University, and this is focused on RNA vaccines for cancer. So here's the talk, here's the agenda. I'm going to spend a little bit of time giving you some basic biology and chemistry and just background on the COVID virus and how it infects people. Then talk about the landscape. There's a lot of vaccines out there and coming. RNA vaccines happen to be the first that have been reached approval, and really talk to you about why that is and why are they special, why are they different, run you through the data from biotech, particularly the safety data and the efficacy data. So if you do get vaccinated, what to expect. And then just a couple of slides on what we're up to at Precision NanoSystems in Vancouver with our mRNA vaccine approach. It should take about 30 minutes, and then we can spend as long as you need to answer questions. So I think, you know, you've been bombarded with propaganda in the press on RNA vaccines. And, you know, I used to live in my own little world a few years ago. Nobody knew what RNA was. Nobody knew what a lipid nanoparticle was or an RNA vaccine. Now the entire world knows, the secret's out. But here's the basic biology. You know, our DNA encodes our genome. It's a double-stranded, and it's very different than RNA. We'll come on to that in the next slide. But the central dogma of biology is our genetic code is laid down in our DNA in cells. And when we want to make something and have function, we make RNA. That RNA is a message. It engages the ribosome, and from it, it encodes a structural protein that's expressed, and it could be an enzyme, it could be muscle tissue, has various functions. So the RNA is just a transient message. If you read some of the stuff on the web where it says these vaccines integrate into the genome, it's just not possible. It's a transient message that encodes a protein from the virus. It only encodes one protein, and the virus contains several, and one protein isn't enough to cause an infection, and it can't go back up into the genome and integrate. So it's very safe, and we'll come on to why is it a lot easier to manufacture than anything based on a protein. The other thing you need to know is a little bit of chemistry, because on this slide, so viruses are made, some viruses are made of RNA, some are made of DNA. So our immune system is hot-wired to detect RNA viruses, and our human body has designed ways to make our RNA look human. And we've introduced base modifications to the RNA structure. Here's one just shown here. This is called pseudouridine. The virus doesn't contain this, but our RNA does. And so Moderna and Pfizer BioNTech have used these mammalian base modifications in their RNA vaccine, so to make them capable of decoying the immune system to some degree and functioning better than viral RNA, making them different from some of the other RNA vaccines that you're going to see in the future. RNA is very different than DNA. DNA is a double helix. It's very stable. It's capable of self-repair. RNA is unstable. It's designed to hydrolyze. It's designed as a transient message so that off your genome, you can make a piece of RNA. It only lasts for up to a few hours or days, and then it's hydrolyzed away and gone. And it's hydrolyzed because the sugar molecule ribose is different than deoxyribose found in the DNA molecule. And that's why you're hearing stories about stability issues with RNA. Needs to be stored at very cold temperatures. It's because it was designed to be unstable and degrade quite quickly. So here's a little bit of biology around the SARS-CoV-2 virus. It's a coronavirus. It's what's known as an envelope virus. So you have structural proteins in the envelope of the virus with the genome on the inside. It binds to lung cells. On the lung cells, you have a receptor, the ACE2 receptor, and the spike protein, which is encoded in all of the vaccines, binds to this spike protein, and this allows it to internalize into the cell. During that process, the virus releases the RNA genome. It engages with the cellular machinery, the ribosome, and it starts to produce more viral progeny. That spike protein goes onto the surface of the cell. It makes more copies of the genome, and those bud off and go on to infect other people. Our immune system will detect this virus. It will chew it up, and you get two types of immune response. You get a B cell or an antibody response, and these antibodies bind to the spike protein and inhibit this infectious process, or you generate a cellular response. These are T cells that will recognize an infected cell with these viral proteins on the outside, and it will kill that cell. All of the RNA vaccines that are currently licensed, the one from Moderna, the one from Pfizer and BioNTech, work through neutralization using antibodies. They bind to that spike protein and inhibit it, binding to the ACE2 receptor. So what we've seen for the pandemic and the response from the biotech and pharma industry has been pretty unprecedented. We've seen an accelerated approval process. And so what we saw from Moderna is on day 0, the virus was sequenced. They sequenced this spike protein. They went through a process on the computer of bioinformatics and produced a piece of RNA encoding that antigen. They formulated it into a delivery system and put it into vials. And so within 45 days of the sequence of the virus being identified, they have material ready to go into clinical trials. By day 66, they worked with the regulators and they were allowed to start vaccinating people. And then in less than a year, they conducted all of the clinical trial stages, phase 1, 2, and 3, and generated enough safety data in over 18,000 people to warrant an approval process for pandemic use. And so we've never seen this in the industry before. Traditionally, these are the timelines that you would see for vaccine approval, anywhere from 100 years to around 10 years. And so RNA really was the first vaccine to be approved, but there are a lot more types of vaccine on the way. On the left, we have what all the vaccines in preclinical studies before they're being administered into humans, and over 100. And over here, we have everything from the World Health Organization in September that was in clinical trials, viral vector vaccines. This is the Oxford vaccine being commercialized by AstraZeneca, protein subunit vaccines. We have a very good one from Novavax, but there are 6 RNA vaccines being used. And so the question is why? Why are we seeing such a large increase in the number of RNA vaccines? And so on this slide, I'm just depicting all the different types of vaccines. And really the difference with RNA is you do not require cell culture to produce the vaccine. Probably doesn't mean a lot to you. So here's a picture. This is the influenza pandemic facility that was built by Novartis Vaccines in Holly Springs. It was a joint venture between the US government and Novartis at the cost of $1 billion. It's close to 500,000 square feet, has over 500 employees. And up the top here on the right-hand corner, you can see the fermentation train. So this virus is grown in mammalian cell culture. These vessels are 2,000 liters each. And downstream from this is a large array of stainless steel purification equipment. And all of this needs to be validated, cleaned, and multiple production runs. And so this facility was designed to produce 150 million doses within 6 months of the declaration of a pandemic. It's designed for flu vaccines only, and it couldn't produce a different type of vaccine. So RNA is different because you don't need cells. It's what's known as an enzymatic transcription reaction. You encode your RNA in a piece of DNA. You add a T7 polymerase And in a sequence-dependent manner, this polymerase reads the code of the vaccine and incorporates bases in a sequence-dependent manner and generates the mRNA. And the enzyme keeps going back and circling. And so for one copy of DNA, you can make thousands of copies of the RNA. There's no cells, there's no cell debris. You can grow these, you can generate this RNA in bags, disposable bags within hours. The process is very clean and quick to produce the vaccine. And so in this facility here, we could probably produce 25 different RNA vaccines. The process for making RNA is sequence independent. So we could produce an RNA vaccine for flu, an RNA vaccine for rabies, one for COVID, one for Ebola, and we could use the same process. So it's very unique, very different, and at a much smaller scale than the pharmaceutical industry has ever seen before. And so that's why they were able to produce these vaccines far quicker than any of the protein-based systems that will certainly come later and be as effective or maybe even better. RNA vaccines aren't probably going to be better than the viral vector vaccines or the subunits. They're just simpler to produce and much faster to produce because the protein is encoded in the RNA and the manufacturing process is independent of the protein. The moment you have a protein-based vaccine, every process is different and it takes time to scale it up and work out how to manufacture the vaccine effectively and at scale. So we've seen an explosion in the RNA vaccine field. I'm not going to run into any details. This isn't new. Over the last 5 years, we've had multiple clinical trials. I'm going to run you through, the technology from biotech, and you can see they've had several vaccines in clinical trials previously, and the same with Moderna. And so the regulators have seen this technology before, and they've seen the safety profile, they've seen the efficacy with other types of vaccine. And there's been even a lot of academic research with RNA vaccines over the last 5 years. So relatively speaking, there's been a lot of activity. There's just never been a commercial product. And the US population really hasn't been aware of this technology, even though it's been up and coming for the last 5 years. So here's the landscape of the RNA vaccines. And so what we've seen is in the middle here, the base modified RNA from BioNTech and Moderna in the front line of the news, but it's not the only type of RNA. Here, they encode those mammalian base modifications. This allows increased protein expression of the antigen in the cells once injected, and it doesn't look like viral RNA, so you can dose higher doses. And so Moderna's dosing 100 micrograms, BioNTech's dosing 30 micrograms. On the other hand, we have a German biotech company, CureVac. This is wild-type viral RNA. They're dosing around 7.5 to 10 micrograms in their current phase 3 clinical trial. So lower doses, but it's potentially the side effects could be worse in a dose-dependent manner. Over here on the right-hand side, this is the area that I specialize in, self-amplifying RNA. These are much, much larger. Typically, a base-modified or unmodified RNA is about 3,000 to 4,000 bases. Self-amplifying RNAs are modified genomes of an alpha virus. They're much larger, 12,000, 13,000 bases, and they're capable of self-amplification. And we'll come on to that towards the end. Much lower doses, so a much larger manufacturing capacity from these. Typically, we're expecting these to work in the 1 to 10 microgram range. Sorry about the dog. So we've seen the Moderna and the BioNTech vaccines being authorized for emergency use, but CureVac is close to licensure in Europe, and we have two self-amplifying RNAs coming along, one from Imperial College London, just starting to finish up phase 2 clinical trials, and there's a local company called Arcturus who are doing clinical trials in Singapore, and they're about to go into phase 3 efficacy trials. So typically for the vaccine process, phase 1 is all about safety. You usually dose escalate, so you start at a low dose and you escalate up to see where there is the adverse event profile starts to get intolerable in the patients, and you look at where immunogenicity starts to occur and potential protection of the vaccine. Phase 3 is all about efficacy. Do you see protection and safety? And we'll go through the data from from BioNTech and Pfizer that they published quite recently. All of these RNA vaccines need to be delivered, and they're delivered in what's called a lipid nanoparticle. So this encapsulates the RNA, it protects it during the delivery process. RNA is rapidly degraded by RNase, and when you vaccinate intramuscularly, you upregulate RNase without a lipid delivery system to protect it and facilitate the delivery process. These vaccines are ineffective. They're nanoparticle size, so they're in the 150 nanometer size range. This is about the size of a virus. The lipid envelope is made up of multiple components. The key component is this ionizable lipid. It's positively charged. The RNA is negatively charged, and that allows it to interact and encapsulate the particle. So this is a slide I stole from BioNTech, just showing the process. So here's the RNA vaccine. It's a lipid particle. The mRNA is encoded on the inside. You dose it intramuscularly. At the site of injection, cells are transfected by this lipid delivery particle. The mRNA is delivered into the cytoplasm of the cell. It engages the ribosome. The ribosome reads the code and it produces the protein. This is then secreted through the secretory process of the cell, and it ends up on the cell surface, just as though you had a viral infection with SARS-CoV-2, and it's folded into that trimeric structure that you would find on the virus, but there's nothing else there. It's just the spike protein. There's none of the other proteins associated with the virus, and there's no viral genome budding off with it. And so the immune system sees this protein and it takes it up and it processes it and it lays down an immune response and generates both an antibody and to some degree a T cell response against it. And as I'll show you in the subsequent data, those antibodies are capable of binding the receptor binding domain on the spike protein and inhibiting viral infection. So that's how the mRNA vaccines work. So you're just encoding the protein. You're not actually manufacturing it. You're vaccinating the subject, and then we're manufacturing the protein, and we're teaching the immune system what this viral protein looks like. And then our immune system is generating antibodies that are capable of neutralizing the virus when we could get infected. So here's the phase 1 data from Moderna. I'm not going to go through this in any detail other than to say multiple groups, multiple vaccines were tested, but very small groups were tested. So we're looking at about 18 subjects per group on the doses that they ended up testing. The vaccine that they took forward is here. It's the B2 vaccine. They tested it in 18 to 55-year-old cohort, and then in a more elderly cohort. They did a dose escalation. So they did 10, 20, and 30 micrograms. And in this graph, we're measuring the antibody response. So antibodies that bound to the S1 subunit of this viral protein. And what you can see is they're vaccinating at day 1 and following up with a booster vaccine at day 21. And you can see that the antibody responses are coming up and they're measuring out a titer, in the thousands. Over here on the far right is a control group. This is human convalescent sera. So these are people who had infection and recovered, and this is their antibody response. And so what you can see is the 30 microgram dose is what they took forward into the efficacy trial. And in the younger cohort and the older cohort, you're generating antibody responses at the levels you see in people who have received and recovered from a natural infection. The most important assay here is the neutralization assay. Here, virus is, you follow virus infecting into cells and see if you can inhibit infection. And we're looking at what level we can dilute the antibody sera that we've collected from patients, and we get a 50% inhibition titer. And we're looking for titers in the hundreds. And again, what you can see is at the 30 microgram dose, we can see day 1, day 21 is when they get the boost, and you can see titers are starting to come up. And we see the same kind of response in the young cohort, in the elderly cohort. Not much of a response early on, and we'll come back to this when we look at the efficacy. So certainly after the first vaccination, there is a time period where people are vulnerable to get infection. But after about 12 days, that vulnerability is gone, the titers are low, and then by about day 28, titers are coming up very high, and they're at the levels of people who have recovered from seroconversion. And these antibodies are binding in here on this spike protein in the receptor binding domain and inhibiting it binding to the ACE2 receptor so it can't internalize and infect the cell, and then lead to amplification and in fact subsequent infection into other people. Probably most relevant to all of us is we're going to get vaccinated. So what is it going to look like? Is it going to hurt? How did the people respond during this clinical trial? So they're getting 2 doses, 3 weeks apart. Half of them got a placebo, the other half got 2 vaccinations. Over 40,000 people were vaccinated. So 18,000 people got the vaccine, and the safety data was generated in over, it was 8,000 patients. You can see it here. And so there are 2 cohorts. You have the younger cohort, the older cohort. This is after the first dose. This is after the second dose. And we're looking here at local reactions. So at the site of injection, it's usually in the deltoid muscle, and the major adverse event was pain at the injection site. They're grading it mild, moderate, and severe. Moderate means it inhibits daily activity. You might not want to row. Severe, you're definitely going to be in bed, resting up and not thinking about getting in that boat and rowing. And so what you can see is, over 80% of patients in the younger cohort were experienced some kind of local adverse event. These were transient, and people went back to normal within 24 hours. The other type of adverse event is systemic, and so these would be things like fatigue, headache, chills, muscle pains in sites other than in the deltoid at the site of vaccination. And what you can see is incidence of in the younger cohort after the first dose and after the second dose. And you can see that there are some severe events, and this would have meant, elevated temperatures, fatigue, headache, and these people would have been laid up in bed transiently for 24 hours. Most people were mild to moderate. How does this compare to, say, a seasonal vaccine? A little bit more severe. It's more similar to the symptoms you'd see for a shingles vaccine. But certainly safe. And this this adverse event profile is showing that the vaccine is working. Our RNA system, even though these vaccines contain base modifications, this is a lipid particle about the size of a virus with RNA in it. Our immune systems are seeing it as what it thinks is an RNA virus. It's not, because it doesn't encode all of the viral proteins. It's not capable of infection or spread, but our immune system is hot-wired to detect foreign RNA, and it's a response to that, and that helps our immune system amplify the antibody response. So this is in the older cohorts, and so less people are reporting adverse events in the elderly cohort. This is pretty normal. Young people tend to squeal more during vaccination. But still, a significant amount of adverse events. You're going to know, the majority of you who get this vaccine will know it. It's transient, 24 hours, and you know the vaccine's working. Here's probably the most important data. Here's the efficacy data. So anything with a solid, either square, that's the placebo, means that's a hospitalized patient with confirmed infection. An open square or an open circle means a PCR positive infection, but it's asymptomatic and not a case of severe disease. And what you can see in the vaccine group, we're coming back in with the booster at day 21. And so the titers are coming up, as we saw in the phase 1 data. And at some point we're protected, and very few people get infected after about 12 days. And you can see it here in the zoomed in area. And you can see lots of severe cases occurring within the placebo population. For Moderna, there was no case of severe disease. With the BioNTech vaccine, there was just one case of severe disease. And so the majority of patients were protected after about 12 to 14 days. The 95% efficacy comes 2 weeks after the second vaccination. They then take a cut and look to see who is protected and who isn't protected. So there's the efficacy data. Obviously, the vaccine trial was finished after 2 months, so they didn't read into what happens after 2 months. They didn't look to see if the vaccine protected against asymptomatic infection and transmission to unvaccinated people. So that's to answer your question, Randy. We don't know if you could get infected asymptomatically. You wouldn't go get tested, and you wouldn't know if you passed on the virus to somebody else. That's why they're still recommending people wear masks, even though they've been vaccinated, until we get past that herd immunity where the virus can't spread anymore. And they don't know how would you deal with people who didn't get the second dose. So still lots of unknown questions. I'm just going to quickly finish up. Obviously, I'm still active in the field. I left my job in San Diego in April so I could go work on COVID vaccines in Canada. I actually haven't been in Canada since March. I've been working remotely, building the team. We've written grants. We got funded by the Canadian government. That funding came through in late October, November. And we're working on self-amplifying RNAs. And so they're a little different. They're able to amplify in a cell. So one copy of a self-amplifying RNA will become 10,000 copies. We produce viral protein for much longer periods of time. And so these vaccines work at much, much lower doses. So from the same manufacturing footprint, we can produce much more clinical material. But this technology is still, unvalidated clinically, and we'll begin to validate it. That facility will hopefully be ready in Canada and validated for the next pandemic. Precision NanoSystems specializes in those lipid nanoparticles. It specializes in making microfluidic mixing devices to make those lipid nanoparticles. here's just the first data. We're collaborating with Imperial College London at showing that neutralization titer up in the thousands. So our vaccine's working in the early preclinical models. Hopefully we'll be vaccinating Canadians in the June timeframe. With that, I'll just say, RNA vaccines have arrived. They're not perfect yet. There's still probably another 10 years of research, and we'll be developing, better safety profiles. We'll get them looking more like the seasonal flu vaccines in terms of their adverse events. We'll be improving the way we manufacture them so that we can produce more material from the same manufacturing footprint. You'll find RNA vaccines for other infectious diseases. I'm working on stuff in the cancer field, particularly metastatic breast cancer as our first opportunity. Also working with people like the Gates Foundation for the developing world. This is a very cost-effective way of producing vaccines. So with that, I'll shut up and I'll hand you back over to Dan.
Well, thank you, Andy, for that. We've got a number of questions from people. First of all, from Cindy Kavanaugh, how long will the vaccine last? Will we have to get revaccinated every year?
Well, right now, Moderna's saying at least a year. So, people who've been naturally infected with COVID, they're showing immune responses that they've measured them out to 8-9 months now. They're still there, and the memory is laid down, and they can be recalled. So you'll be protected from that virus, but obviously viruses mutate. This one is mutating. All RNA viruses mutate, and so how long we'll get protection before the mutations outgrow the vaccine, we don't really know. It's probably going to be a few years, but right now the prediction is at least a year.
Okay.
But would you, once you've got protection to one coronavirus, the likelihood of severe disease from another coronavirus, it's like flu. You start to build a repertoire against all the viruses. So we'll just have to see. It's an unknown, but we can certainly generate the vaccines as fast as the virus mutates now.
Okay, Sue asks, are you saying that you need about 7 days after the second dose in order to be fully protected?
Yes, but you do have partial protection probably after about 12 days. There's certainly no incidence of severe disease after about 12 days after the first vaccination. So there's partial protection, but you could still get infected. But would you be hospitalized? It's highly unlikely. And certainly as you get out, as the titers go up, you get protected.
Okay. Kate asks, can you transmit the flu if you have the vaccine?
So can you so when she says the flu, she means the coronavirus?
I'm not sure.
Okay. So flu and coronavirus are two different viruses. So this vaccine, the COVID vaccine, will not work against flu. So you could still get infected with flu. You would get flu, and if you are coughing and spluttering and infectious, you could be spreading flu. If she means, could you spread coronavirus if you are vaccinated? Potentially, yes, because those titers will drop. And if they drop down below the threshold where you see protection, you could get infection. Your immune response responds. It has the memory, and you will clear the virus, and you won't get severe disease. You may even just be asymptomatic with very mild disease, but you could potentially have replicating virus that you're coughing out and infecting people who are naive and who have not seen the virus or not been infected.
Okay. Scott asks, why is PEG suspected as the cause of anaphylaxis?
Because there's a lot of pegylation happens in the drug industry, particularly around antibodies.
Can you say what PEG is?
Polyethylene glycol. And we are exposed to polyethylene glycol. It's in toothpaste, it's in shampoo, it's in quite a few pharmaceutical products that are administered intravenously. And so some people do have an immune reaction to PEG. So these lipid nanoparticles contain the outer coat of it. It is a very small molecular weight polyethylene glycol. So it's suspected that has caused, adverse events it's about an incidence of about 1 in 100,000. There's still no published data yet to confirm that it is the polyethylene glycol causing the problem, but it's suspected because there is a history of people having allergic reactions to polyethylene glycol. Those people have been administered an EpiPen and recovered and been pretty uncomfortable for 24 hours.
Okay. Sue has her hand up, so I'm going to allow her to talk.
Okay.
Hi, Sue.
Hi, Sue.
Hand up. You're muted, Sue.
Sorry, it was a mistake.
Oh, okay.
You answered my question already.
All right.
Okay.
All right. Richard had emailed a number of questions.
Of course he has.
I'm going to turn this over to Richard for a minute. Richard, are you there?
I think you've already answered the question, thank you.
No, not you, Sue. Richard.
I'm here. Can you hear me?
Yeah.
Great. Thanks, Andy. Thanks for doing this for everyone.
No, you're welcome.
I just had a couple of questions about the mRNA vaccine efficacy for some of the strain mutations we're seeing. And the concern is obviously for those who have higher infectivity or severity of illness. I've been following it. There have been a couple just within the last week, that journal bioRxiv released a couple preprint small studies, in vitro studies for the Pfizer vaccine with regard to the UK and South African variants. One used blood from vaccinated subjects and the other one from convalescent serum. I'm just wondering, what's the fastest and most reliable way that one can assess whether an mRNA vaccine is going to be efficacious when a new variant like this appears?
I mean, right now, Richard, we don't have a correlative protection. So we're assuming it's that new titer, but that's only one arm of the immune system. There is a cellular response for the Moderna, Pfizer, BioNTech vaccines. It's not stellar. Certainly for the self-amplifying RNAs, it's much more potent. For the unmodified, it's more potent. For the viral vaccines, that's their main mode of action. And so they may be fully protective against all of these mutations. And we'll just have to wait and see. But it certainly looks like most of the variants are covered currently. The South African one, there's just been one study where they did some in vitro work and showed there might be a problem, but a lot depends on where did the sera come from? How did they, process it? How did they apply it? What concentrations? There's lots of caveats, and those journals are not peer reviewed. So, I think we'll just have to wait and see. But today it looks pretty good. It looks like it's fully protected. If you're really worried, to me, probably the most potent vaccine coming is from J&J. They have an Ad26 viral vector vaccine that looked fantastic in the primate challenge models. And it gives you both arms. It gives you antibody and T-cell. And those antibodies, let's wait for the data, but they're probably going to be higher in magnitude than than Moderna, BioNTech, Pfizer, and plus they have the T cell side too. And the T cell is recognizing, peptide maps of the protein, so it's less dependent on the mutations, because it's looking at fragments of the whole protein. And nobody's measuring IgA. IgA is like the mucosal antibody, and that's probably more predictive of what's going on in the lung mucosa.
Interesting. I've seen some comments in the lay press that if we do have to modify an mRNA vaccine because of a mutant, that it's pretty quick, like 6 weeks or something like that. Is it really that quick to develop?
It depends on the regulators. I mean, it's ready to go in 6 weeks from a manufacturing point of view, because
okay.
The process is sequence independent. So it's really quick, but a lot depends on how the regulators are going to think about, is this a new trial? Do we even need to do a trial? How are they going to handle? We saw, Moderna were the first in. They had no GLP tox. They did the GLP tox in parallel to doing the dose escalation in phase 1. Because they vaccinated, they had 5 or 6 clinical trials prior to this, where they kind of validated the platform with the regulators. We certainly haven't done that with Health Canada with our strategy. We're doing the first one real time, and we'll be building out our data set with them. But a lot depends on the regulators and how they view the technology.
And that actually, you anticipated my third and final question. That is, if what would the FDA require for an emergency use authorization if there was a modified RNA vaccine, a modification based on one they've already authorized? And would it be a clinical trial, or could it conceivably approve without clinical data?
I think my guess is that they would require a very short clinical trial in 100 patients, whatever, where they collect sera and they show a correlate. They show that they can neutralize this virus relative to other isolates.
Yeah.
And they do a cross comparison, so, but I'm not at the FDA.
Yeah.
And it's like having a crystal ball sometimes on how they view things.
Yeah. All right. Thanks for those answers very much.
Yeah, great questions. Thanks, Richard. Okay, I have some more questions from Scott. Why vaccinate patients who have been infected and recovered?
Because their protection may be suboptimal. And so, antibody titers do wane over time. And so you can, you saw the spread on that human convalescent sera as the control. So some people have very low neutralizing titers. And if you vaccinate them, you'll boost them and they can't get infected anymore. Whereas if the titer is low, they could get infected, they could be asymptomatic and be capable of spreading a virus without people knowing. So that patient isn't in danger, but they could potentially, be spreading infection to those that are more vulnerable.
Okay. Scott also asks, is there a work in progress on a universal mRNA virus against all strains of the flu to avoid annual vaccinations?
Absolutely.
I'll ask a follow-on question. Will we have it next week?
No.
Is that one of the things that's on maybe a 10-year timeline?
It will be on a longer timeline because, seasonal flu isn't a pandemic. There's, the risk-benefit ratio is different. You're talking about vaccinating, a healthy population against a lower potential risk. But, it's coming. People are working on it. I've got plans, at the company in Vancouver to be working on those universal flu vaccines. We believe we can design antigens that will be more conserved and capable of producing a broader protection against a whole range of different flu viruses. But clinical trials are going to happen for those over the next couple of years. And it'll probably be about 5 or 6 years before we see anything licensed at the fastest.
Okay. Alexi asks, can you take several vaccines over a period of time? And I'm going to read between the lines here. If you don't get the second vaccine within the 21, 28 days, do you have any comment on that?
It's fine. You can vaccinate later. Obviously, if the titers come up and come down, you could be vulnerable. But optimal for this type of vaccine, Moderna biotech, is 21 days. A self-amplifying RNA, the one I work on, optimal is 12 weeks, 8 to 12 weeks, a much longer interval. You get a much better boosting of the immune response. So, if you go late, it probably isn't going to be detrimental, but there is a period of time where you could be vulnerable to asymptomatic infection. You don't know you're infected. You could be shedding virus and infecting others. That's the danger.
So is there, if the choice were to wait for the self-replicating mRNA?
No, there is no choice. Your choices will be the 2 RNA vaccines currently. You've got the Oxford vaccine getting licensed, and which is a viral vector system. There will be the J&J vaccine, which is preclinical data was certainly the best vaccine. Single shot, you only need one. But we don't have the efficacy data yet. It's probably about 2 weeks away. I think that'll be the best data. But I'm a scientist, we need the data. We'll just have to wait and see. So there's certainly options out there for everybody.
Okay.
But the self-amplifying, no, it's not going to be ready this time.
Okay. All right. Melanie asks, even though it's early in the whole process, are there any known conditions a person could have that would indicate they should not get the vaccine?
Well, emergency use authorization was given based on the clinical populations that they tested in. So they did not go into the immune compromised, pregnant women, younger people below 18. So there's a whole range of stuff that isn't under that emergency use authorization. But obviously physicians can make risk assessments and recommend vaccination. This is a new technology, the RNA vaccines, and the more traditional vaccines are coming. They have a much more established safety profile. And, so maybe, in the immune compromised, we'll start to see physicians using those. But, I'm guessing that Pfizer and biotech and Moderna are testing them in these more vulnerable populations in extensions of their clinical trials to try and generate data. But, because it's a new technology, they haven't branched out into those more vulnerable populations yet. And so they're not going under the emergency use authorization.
Okay, Randy asks, can you discuss the argument for getting vaccinated? I hear some people who are hesitant to get it, stating that it is unproven, etc. I guess they are in the same types who won't get a flu shot, but this seems very different.
Yeah, I mean, so I have interesting discussions all the time with Uber, cab drivers on this subject. So I'll just give you a scenario I usually give to the Uber cab driver. When we start talking, they find out what I do. And, they tell me, well, I don't believe in vaccines. And I say, okay, so here's the scenario. Young kids are vulnerable to infection and death with influenza. And so are the elderly. So you have, elderly parents in their late 80s, and your son is married and you have an infant who's a month old. You get flu, either of those sets could die. Can you live with that? Because I'm telling you scientifically, you could be protected and you could have inhibited that process. It's your decision. You need to make up your mind. But that's, you know, that's the reality. You could be looking at, I could have done this. Scientific sense is there. You will see protection and you could have prevented that death. And it comes home because it's personal now. It's about people you know.
Okay, thank you.
But Randy, all I can do is give you the science and allow you to make those decisions.
Okay, I don't see any hands raised. I don't see any more comments in chat. Does anybody have any additional questions for Andy? Ooh, hands are up.
Oh, Pat Hayes.
Pat Hayes.
Hey, Andy.
Hi, Pat.
Well done.
Thank you.
Cynthia's here listening as well. Thank you very much for your time.
Oh, no, you're welcome.
So a couple of questions. I'm not sure they're going to be world-solving questions here. When you're looking at the data for dosing for all these different age groups, was the same dose used on all age groups?
Yeah, I mean, they in the phase 1, they did a dose escalation the same as in the younger cohorts. And they saw very similar data. And they went with the same dose in both the young and the old. The safety data profile looked better in the older population, but it may just be that they grumble less and they just say, it's, you know, it's fine.
That's true.
Well, that's probably true.
That's probably true. But, you know, it certainly produced protection and an immune response in the elderly, which has not been the case sometimes with protein-based vaccines, and we've had to up the dose in the elderly.
Right. The UK seems to be on a track where they want to vaccinate more people with a single dose because they can hit a broader swath of population rather than necessarily get to the second dose. Can you comment on what the implications are of that?
Yeah, I mean, you know, so they're looking at trying to provide partial protection. Well, so they're going to inhibit severe disease and hospitalization. There may be, you know, infection, but it's asymptomatic or it's not resulting in hospitalization. So they've prevented the overload of the healthcare system, and then they're going to come back in and vaccinate with the second vaccine. And with the Oxford vaccine, which is the viral vector, 12 weeks is fine with that vaccine to come back in that late, because you can boost responses quite dramatically. So they're just realizing that they can't vaccinate the entire population that quickly and get 2 doses in.
Yeah. Last question, you mentioned this J&J virus, J&J vaccine.
Yeah.
It seems like at least with the US approvals that this technology, the RNA type vaccines, has kind of cracked the code, meaning that the path to approval, I know it's been a pandemic and there's been a lot of pressure to make it happen. Do you think this is going to open up things a lot faster for that vaccine or other ones that you mentioned?
Yes. And certainly there was a guy called Geall who published in 2015 a paper called Vaccines on Demand based on RNA that predicted all of this. And I'll send it to you. Because I was lobbying the US government back in 2013 to build a pandemic response facility based on RNA. And $400 million it would have cost them. And they said no.
Oh my God.
It was kind of a softball question. We'd had that discussion.
Yeah, I know.
Thanks very much.
Just for those of you know, I went to the Defense Advanced Research Project Agency in 2011. It took a couple of years, but at Novartis, I got us $28.4 million to fund RNA vaccine initiative. And over the next 5 years, we built out the platform. We worked with the US Army on Ebola vaccines, with the NIH on flu vaccines, RSV, and we were ready to go, but the government didn't fund us. Novartis Vaccines was sold and asset swapped to GSK in 2015. I left at that point, came back to San Diego to continue my rowing career and enjoy the sunshine. But I've continued to work on RNA vaccines, and it's great. It's mainstream now. I think it's here to stay. We were lucky though, Pat. The coronavirus we knew a lot about it. 2002 to 2003, we had SARS in the Middle East. Then 2012, we had MERS. Both the coronaviruses, a lot of work done on the spike protein, a lot of vaccine work, animal models developed. So when SARS-CoV-2 came along, it was, a lot was known scientifically, and so we could do a rapid evaluation, and we knew it was a spike protein, and we could go forward quickly. If it was an unknown virus, different story. It's going to take 3, 4, 5 months. We just don't know, because there could be 5 different proteins on the outside of the virus, and you've got to figure out which one of them is the important one. Which one is going to provide protection? Do you have to do all 5? Can you get away with 1?
Thanks, Andy.
Hey, Pat, I had a couple questions forwarded to me that you emailed in. Did we answer all of those? Your questions were, does the dose vary by age? How important is it to receive a second dose? And any guess as to when the J&J vaccine will be approved?
Yes, I think Andy's asked that.
Yeah,
pretty well.
Approval, we don't know, but I'm expecting the phase 3 data in a couple of weeks. And I'm reading all of the papers as they come out and kind of documenting them. And I'm giving seminars all over the world on Zoom, trying to educate people on all of the technologies coming. RNA viruses are working, but all the others are going to work. The spike protein is a great antigen, and all the others are going to work too, just fine. There's nothing magical about RNA. It's the spike protein that's magical, and we just got it's immunogenic. Our immune system knows what to do with it, and we generate those neutralizing antibodies.
My dear wife Cynthia had a question.
No, I'm good. He answered it.
Okay, we're good.
Thank you, Andy.
Anything else, Pat?
All done here.
All right, thank you. Evan asks, does the J&J vaccine use the same viral vector or type of viral vector as the Oxford one?
Broadly speaking, yes. The adenoviruses, but the J&J one is a human isolate. It's an Ad26. It's very, very rare. And so there's no pre-existing exposure in most of the population. So it works. The Oxford vaccine is a chimpanzee adenovirus. So it's only in the chimpanzee population. They isolated it. So the problem with these viral vector vaccines is the outside is protein. And so if our immune system's seen it before, we already have neutralizing antibodies. And so they won't work if we've seen it before. And so they have to go either to a rare human variant, or they go to chimpanzees where we haven't seen it before.
Okay, I don't see any other hands raised or any other questions. Please, everyone, feel free if you don't feel like your questions have been answered. I'll give you 30 seconds here to tell us that you'd like to hear more before Andy can close out.
I can go get my glass of wine.
Well, you know, giving people goals is a good thing. Ask your question within 30 seconds.
Oh, here we go, China-Russia vaccine. Sputnik 5. Yeah, so Sputnik is an adenovirus, but they've come up with a prime boost, so they have two different isolates, two different types of adenovirus, so that they can prime and they can come in with a different outside of the virus to boost. China has about 4 or 5 different vaccines now. So they have a live attenuated coronavirus, so it's the whole coronavirus, but it's been so weakly attenuated, it's barely capable of replication in humans, and they vaccinate the whole thing. It kind of weakly amplifies and then fizzles out, and it generates an immune response. They also have a split viral vaccine, so they grow the virus in cell culture, harvest it, and then they kill it, split it, and take the outside envelope, and then they inject that in. They also have an adenovirus as well system. I think it's an Ad5 vector. Doesn't work particularly well. And then they've got some homegrown variants of RNA vaccines, but they're very early in the clinical pipeline. None of those vaccines are coming to the US. You know, you've got, you know, the 2 RNA vaccines. There'll be a protein subunit from Novavax with an adjuvant, and we'll see the J&J vaccine. I'm not sure that the Oxford vaccine's coming across. I don't know if Warp Speed put in a purchase order for it. Okay.
And here's another question. Any comment on the Chinese and Russian vaccines?
I haven't been tracking them particularly well. Certainly, some of the Chinese vaccines aren't working that well from the data that I've seen. Don't know much about the Russian vaccine at all.
The Chinese vaccination is working or is not working based on
some of them are, some of them aren't. There's 5 different types. So yeah, some of them work, some of them don't. But there's not that much information on them. In the same way there are on you know, anything from the Western, from Europe and the US. You know, what we're seeing is, first of all, publications come out in these online non-peer-reviewed journals, and then they're followed up by a more extensive analysis in a peer-reviewed journal, you know, such as New England Journal of Medicine, or in Nature, where you know other scientists have reviewed and been through with a fine-tooth comb. So you know the data's traditional, thorough, and it's very timely. I've never seen data coming this fast out from programs. So they've been very open in communicating with the world and conveying the data and allowing scientists like myself to see it almost real time.
Okay. I don't see any other questions or hands raised. Last chance, everybody. Talk with a medical expert. All right. Andy, I very much appreciate your time. Any closing thoughts?
You know, hopefully I've encouraged you to go get vaccinated. You know, and the sooner we get herd immunity, the sooner we can put this all behind us. You know, be sitting around sharing stories and medals and trophies.
I look forward to that as well. Okay,
Absolutely.
Andy, thanks for your time this evening, and for all of the attendees, thank you for being patient with the pushback of an hour. Appreciate that. And this presentation was recorded and will be put up on YouTube, and we will email and post the link to the YouTube video if you'd like to rewatch this or share it with others. And Joanne says, thank you so much, Andy. Miss you and Allie. Tom Cook says thank you. Cindy Cavanaugh says thank you.
Oh, you're welcome, everyone. I look forward to coming back. Can't wait. I'm getting tired of the yoke.
All right. Okay, thanks everybody. And that ends our presentation for, whoops, I've got a question.
Oh, there
It's
you
a
go.
thank you from Randy Hanna. Okay.
I'll be back, Randy.
Okay, so this ends our presentation for this evening, and thank you all very much. And I hope to see you at the rowing club and on the water. See you soon, Andy. Bye-bye.
See you soon, Dan. Thanks, everyone.